Okay, we are already aware what is gut microbes. Right? So, in one dozen gut microbes are also known as gut microbiota. It actually presents in your colon. So, the body normally has like trillions of gut microbes are present that could be in both the beneficial bacteria as well as harmful bacteria. And the gut system is also called a secondary brain as it plays a major role of your entire immune system. And talk about the gut health that is important. So a healthy gut will actually help you to maintain a balance, and also it acts as a strong barrier. So in case of any inflammation or cancer, these beneficial bacteria will actually help to combat all those inflammations. And so what disturbs this gut health because of the lack of fibers and improper diet, stress and toxins when you are undertaking a lot of drug side medication that will lead to toxins and that eventually result in causing the inflammation. The aim is to develop a probiotic lead formation using pomegranate seed and red onion to evaluate its effectiveness in gut health improvisation or maintaining a balance, and also inflammation reduction through bioprocess methods. So the objective is to formulate a probiotic ring and to analyze the bioactive properties of both pomegranate seed and red onion extracts, and to investigate the anti-inflammatory properties which are responsible for your inflammation, and to study the bioprocessing engineering, which is very important in this entire step, and to evaluate the effect of probiotic ring acting in your colon. So why pomegranate peel? Pomegranate peel is actually rich in polyphenols, tannins,
Can
and
you just go to the results? Because these are, everyone knows what is pomegranate peel and red onion. What is your interest? So what you did?
basically this is a startup idea.
Oh, startup idea. Startup idea means there are two ways. One, there is already you have most prebiotic and probiotic drink, which is your rice, overnight rice or water. Okay, that is the best prebiotic and probiotic. So why you are going for this first question? My next question is why omega and P or like that's what you are going to tell and red onion. Why that combination?
I have studied, they have already studied separately for pomegranate seed and red onion. I thought of combining these two because they are more helpful for reducing the inflammation. And also, the main thing to choose the red onions and pomegranate is because it is mainly focused on mineralization. So we may get lot of wastage on the pomegranate peels, and also during the monsoon times, there are a lot of onions that are getting wasted, right? So that is the actual aim, to utilize those waste materials, like literally peels of those components, and to develop a probiotic ring. So it will be very sustainable, eco-friendly, and also there is a major role in valorization.
So why combination? Because you, pomegranate peel itself is a good probiotic. Onion is a probiotic. Why you want to combine these two?
Because of their natural properties like polyphenol and quercetin, there are a lot of properties in the studies have already been proven. So
Red onion. Okay, instead of red onion, if you go for white onion or the shallots, the smaller onion, that is much more prebiotic and probiotic than the red onion. Why red onion? What it contains? So you should tell me what it contains. Why you want?
it has oil and fructose or oligosaccharides and ingredients.
So in the last people also said there is oligosaccharides, phenols, all the plant product or all the plants will have phenols, polyphenols, oligosaccharides. Everything will be in a different combination. So everything will have everything, but in a different concentration. What is the concentration? make a product. So without knowing that thing, you can combine anything because everything is going to grow back again. So what is the optimum and how much is growing or how much is giving is what you need to tell to make a probiotic drink. Because we have the best probiotic drink to my knowledge is the soap water, the rice water. Okay, that it's been fermented and then it's very good. There, if you add red onions to it, it's going to add more probiotics. If you add the whatever, but this is going to give you a sour taste. This whatever your peel of your pomegranate. How you are going to tackle that?
Actually, not all the red onions have a high amount of sulfur. So that is the reason why those onions have a high.
What is the role of sulfur in inflammation?
Sulfur actually helps in those reducing the oxidative stress.
Could you just give me an overview of how that is going to reduce your oxidative stress? Because in a technical term, so it's in a common term is fine. In a technical term.
I'm sure about that in-depth concept.
So saying this, there is something called free radical. There is a free radical. Free radical is the one that's going to cause mutation. So if you are going to, if you have more free radicals, it's going to cause more mutation. So sulfur is the one of the one that is going to control your free radicals. So we can control the free radicals, but that doesn't do anything with your prebiotic or probiotic. So that is going to do for cancer things. Prebiotic, probiotic. Sulfur doesn't have anything to do with your prebiotic or probiotic. Sulfur is good for and like anti-cancer property. That all is there, but prebiotic or probiotic.
Well, actually, I want to reduce the sulfur compound because I thought like audience are having getting that pungent flavor because of a high amount of sulfur oxide. So, I think I have to look onto the methods that could actually reduce the sulfur compound so that the drink will be can be made in a very graceful manner. So, and also we should add some taste enhancers in a natural taste enhancers like stevia or jaggery, some kind of sweets, which actually also helps in your digestion problems also. And then we have to.
So what you have done? What is say you are going to have a formulation? What you have done? One day, like in this formulation. What is your modeling? You haven't started with it. This is your idea.
Actually, a startup idea I have made, so I thought of presenting it right now.
Correct. But two things you need to make it clear. So we are going for two different combinations. What is the best out of it? That all you should first start working out in your technical terms, not in a overview. Overview for general audience. So to get permission from the FSSI or anything, you need to be more technical. You need to tell what is this. These all everyone knows. That the improvement and the information, everything knows. So, but still, why this two? That my main question. So you can combine onion with your rice water. That will be more probiotic. So why you are combining that with your
So like pomegranate is very suitable to begin because it's actually
pomegranate is good, but pomegranate peel will be sour in taste. So no one is going to take that up.
not about only the seed, it will include also the seeds also. The normal juice extract we usually take from the pomegranate.
That is also kind of a pungent or like won't taste sweeter. People need to have it much more sweeter or salty. Then only it is easy to drink because so you can't give it as a milk. It's a kind of probiotic. It has okay, so you know this yogurt, what is flavored yogurt. Why flavored yogurt is selling more? Because that is sweeter. Yogurt is good, but people don't drink yogurt. So if it is a flavored yogurt, that gives more taste. So taste should play a very important role in selling your product. This is all product wise.
I'll work on with the product also.
Yeah, fine. Yeah, these all you need to do.
This is the entire overview of the bioprocess involved in this product formulation. Also, we have to the major means that we have to focus on the bioprocess sector, that what kind of application we are using, what kind of vessels we are involving in this process. So in case the probiotics will always work on anaerobic conditions. So that helps in formulating this probiotic thing. And also talking about the marketing strategy, there are a lot of recently people are.
I'll say finish of that and then you go for marketing, okay?
So here I have stated all the taste, aroma and texture assessments will be that the basis of the consumer's preference. Okay? So we have to take it review or based on the consumer's feedback, and then we have to work on how what kind of formulation we are going to proceed. It could be in form of supplement, it could be form of in powder form, or it could be performed like a probiotic way.
So yeah, these are all we can understand. So I wanted yours of product. Okay, what is the research that you have done for your product? And what is being right? Okay, right. The next again also. So we make it sure that it is 7 minutes. Okay, 7 minutes plus 3 minutes. So it will be much more. So don't need to talk about the basics. Just go to the results, so that I will try to ask you some questions that will be not probed. Okay, we start going to the introduction line, it will be waste of time. So go straightly, go to start to your type, go to your results. So that will give you an idea. Okay, make it to 7 minutes.
Hi everyone, myself Hemanthan, I am from the Faculty of Philosophy, studying third year bioinformatics. Now I am going to say about my research topic is identification of the genes associated with the violent cancer using genes, protein interaction. My intro is cholangiocarcinoma is a very rare cancer and very aggressive way oxidation from the bile transports to the bile from the liver to the small intestine, and later this cancer has increased to 10 to 15% by all over the world, and where it is affected to the many of and the CTA has nowadays for the violent cancer there is no proper cure for the stages like stage 1, stage 2, stage 3, stage 4. But in every stage itself we have small prescribed medicines and chemotherapy, some type of technique. Now I plan to make some genes to the protein where it is mainly affected to the cancer, and where it will be vital role for the pharmaceuticals to produce some of the medicines and something. And with according to my research, the two things are the UDP3 and PYSL. UDP3 where this protein involved in this small unit of chromosomal DNA, RNA, and this UDP3 and PYSL are used for the biogenesis. And nuclear assembly and playing key roles in the cell proliferation. The cell proliferation.
Yeah, you can go to your, what is your research? Workflow of it, not
For my topic, where I have chosen this NCBA-GO database from the NCBA, and later I have where in the NCBA-GO there are three type of data sets: microRNA data and RNA sequence data and methylation data, where I have used the microRNA data with DHC 1054 is a data set name, and later I have integrated this p-value and adjacent p-value like p-value like 0.0001, where before itself step 14 I have made this and not said about this, and I have changed some of the things also, and I have changed to the 0.0001 for the highly confident PETs, and later I have segregated by using the updated and downdated by not R programming, and later I have used this string database for the visualization, and later I have segregated the updated and downdated and by using the string before and logo other objectives. And these are my results and discussion. The first one which shows the Walker plot. The blue color indicates the down-regulated and red color indicates the up-regulated genes. And later these are the network pathway analysis by the cytoscape. These are the total genes present in that cytoscape. And later from these genes I have taken the cytohopper, these are the top 15 genes by using MM6. Means mean neighborhood and mean neighborhood analogy, where it shows the main.
I can understand because so yeah, just tell me what gene is responsible. The best gene.
BYSL and UDP3. These are the gene ontology. The main difference between the fluco, this is the fluco, fluco and fluco where it is called the reactome, where it shows the same pathways with the small amount of the ribosome RNA, and before where it shows the centrality and the cell responses and the remaining things which where cell growth, and then we according to that we will show this, and then geo distribution, p-value and genes are mentioned, and due to the I am writing this research paper, still not complete, I have to extend this properly. And these are my conclusion, like cytoscape, by using cytoscape I have completed this gene, and the UDP and BYSL are the main two genes, and by using cytoscape and global analysis we cover the main genes, and by using after that for the future responses we can use the Apodoptina and the remaining thing for the pharmaceutical drug treatment by using the ADMET and remaining things, and we can copy paste their model.
Good. My question is, you have found out that is UDP genes and BYSL that is responsible for bioinformatics. Okay. So where is these genes present in the cell? Any idea? Because see, you want to target this gene. Okay, whatever the target. So the drug or something should go and from outside to inside. I think I told you last time, or like I told you for some other thing. It should come from outside to inside. So your drug should be either small enough or it should go into the channels and then go inside. How it is going to go, that keep in mind and design it.
Ashna Walla, Meghna Chaudhary Wadila from Savita School of Engineering to deliver presentation on network pharmacology and gene ontology, comparative analysis of shared gut microbiota associated genes in schizophrenia and bipolar disorder. Hello everyone. My name is Pranati. I am from Savita School of Engineering, studying third year bioinformatics. This is my topic, network pharmacology and comparative analysis of schizophrenia and bipolar. Like schizophrenia and bipolar disorder have similar genes. Because they are caused by various factors, various similar factors like disturbance of gut microbiome and environmental factors, diet factors and everything.
Based on some literature review, we have collected some similar symptoms of schizophrenia and bipolar disorder, and then we have the similar gene in the warrior. For materials and methods, we have collected from literature review, and we kept in there is some server called phenolizer. We kept the all phenotypic term in phenolizer, and we have generated this particular network. We don't want in background noise, so we have taken this top seed gene, and then we have constructed protein-protein interaction network. And from here, my lab will continue.
Good morning everyone. So in figure 3 we can see the gene network pathways of the seed genes which are taken from cytoscape, and the cytoanalysis of top 10 genes in which TBL2, TRIN50 and GTF21 are the top 3 hub genes. In this graph, this is a bar graph from gene enrichment analysis. Then we can see that acute inflammatory response and regulation of acute inflammatory response have a log value of 1.4, and neuron projection maintenance have 1.6 value. This is a table taken from gene ontology analysis using shiny protein. In this we can see that the genes are of high level. They have high levels of gene ontology analysis. This is a network analysis taken from switch database, switch tool. In this we can see the
So you can, sorry, you can just identify maybe gene.
the circle we are seeing here. It is a compound of protein or chemicals. This entire tool is the
Is that a protein or chemical?
it depends on the structure. Like the circle
What the name? What the name of that?
stitch tool.
This tool is fine. The protein or the name that
DCDN1.
what is that? Protein or a small
It's a
protein?
protein.
Protein.
It's a chemical and protein interaction.
Yeah, I got it. This is a drug, this is a protein.
This is the drug cell,
Okay.
and this is the protein.
So what is the drug name? What is the protein name?
So this is acetylcholine. This is acetylcholine, and this is SUNGA. These are the different protein interactions with the same. And this is from Shine tool. And these are from this we can conclude that because schizophrenia and bipolar disorder are misdiagnosed in some patients. Based on symptoms that we have taken from literature review, we concluded that drugs can be developed using only the symptoms. Because in some patients, schizophrenia is also diagnosed with epilepsy or also drug-induced depression.
So but that's not with this you can't conclude.
Okay, sir, fine.
But it's good thing. But I have two things. What is gene? This explain what is gene ontology. So because many people doesn't know what is ontology.
The ontology is nothing but biological roles. Like in this particular example, in nucleus, like they have the particular gene is having function in nucleus, mitochondria, something like that.
So oncology is different, ontology is different.
Ontology is different and ontology.
My next question is here you are going to analyze the gene ontology. How come you get the small molecules? So if you are going to work on gene ontology, all the genes will be there. Genes are proteins. Okay, these are proteins. How come a small molecule comes in? So you are working on the gene ontology. Okay, in this gene ontology network, all are proteins. Correct or not? Huh? All should be proteins. How come a small molecule comes in?
These are least important in this network actually. Small molecules are least important and these big molecules doesn't have least structure.
But least important, we should not include these small molecules into the gene ontology. Am I right or no?
Yes sir, yes sir.
Okay. That's what I'm asking you, is it whether the small molecule or of the gene? Because if you are working for gene ontology, with gene ontology, it will be genes. Okay. There is not very small molecules to work. Okay. So yours, this is fine. These are all genes. So you don't have small molecules. So ontology, if you're going for gene ontology, it should be only genes. Correct. I understand what are you doing. Yeah, that's all fine. That's all fine. I understand. You took these genes and then you put it in stitch and then you will get another. In stitch, it should be only gene, not the small molecule. Got it? Either you should do only small molecules or you should do only genes. So small molecules and genes should work for docking.
Docking is done after this process.
Yeah, docking is done before after. Go to previous slide. There's another question. So here, so it is gene enrichment analysis. What is the enrichment that you do?
Gene enrichment in the sense, it's like the particular biological parts.
Right? That's all fine. What is your contribution in the enrichment? If you are saying is gene enrichment, it means already enriched or expressed. Expressed is what in enrichment. So what is your contribution to this analysis? So where you got this data? Minus log FBR.
We have kept our genes in there is server called Shiny.
Okay, shiny go. Okay.
Yeah, we have selected that and we have got this.
So how that shiny go will tell which gene is expressed more, which gene is expressed less? High confidence of what? From string data set. But string data set doesn't give you the expression.
Particular 10 genes. From those particular 10 genes,
See, I'll tell you this. So you have taken 10 genes that I can understand from string data set. String data set, there will be 10 genes inside each cell. So that string data set, whether it gives you what is your expression level or not. Tell you this. If it is not giving expression level, how you can do the gene enrichment? So we know what is the definition of gene enrichment. Gene is expressed. More expressed. That's what I said. Yeah. That 3 gene is more expressed. So least one is not expressed. Only
These
said
parts.
so your gene enrichment analysis.
In cytoplasm, we got chocolate companies. We have taken those chocolate companies, gene enrichment analysis, shiny book. For these,
This I can understand.
These.
This is your cytoscape. Cytoscape. This will give you which is the best gene and these all these.
that we have taken
These all I find. The next slide is what I find. These all I can understand. So, what will tell you the gene enrichment? And the enrichment can't be tell without the expression level. Or you should do the enrichment. So how you do the enrichment? Got my point? So it's not for criticizing or anything. Just I'm asking how we are going to defend this gene enrichment. Thank you.
Sai Irfan and Rohit from Savita School of Engineering.
So I think many people are not understanding what they are telling. Correct? I understand what they are telling, but you people can't understand. What I'll do is, so I'll give you 10 minutes time. You present. Okay? Present panita, I'll ask question. You make sure the presentation is 10 minutes. People are going more than 10 minutes. Adala on the time constraint. I skip the introduction and I'll go after. So you make sure that you present for 10 minutes. 7 minutes, 10 minutes. Then I'll ask two questions. I'm not going to ask any two. So finish the talk so that people can understand what they are doing. Okay? Okay. So it's on. Keep your mic.
Good morning, myself Rohit from Kalka School of Rohit from Kalka School of Engineering, and my friend Irfan. Today we are going to discuss on the topic system biology approach by using the abstract in laryngeal catheter solution. Basically, laryngeal catheter occurs by consumption of tobacco. Larynx is nothing but the voice box which is used to generate voice. Majorly, here we are gonna here Shastriya expression. We are going to see Shastriya expression. What is Shastriya expression means? Shastriya expression is an immunological response. The cancer cells are getting processed in the body. The immunological cells goes to the cancer and makes the cell dead. While the process, the Shastriya expression is more expressed in the T cells of the immune system. By using the Shastriya expression levels in the human body, we are going to predict the laryngeal cancer. So now my friend will tell about the medical system. So my friend will tell about the medical cell methods.
So this is the process we have done with research. So first we have to take collect the data set from the cyto or ECO amoebas, and then we have to normalize the Excel sheet. Then we have to collect the updated and downdated genes. Updated will be we can take up to 60 and downdated also we can take 50. Then we have to input the string database for string interactions. So then by using cytoscape, we can find the top genes. Next, by using Google, we can know about the gene function. Then for docking, we can take a protein and ligand from the PDB and DDBR. So by using Pyrex, we can do docking, and by visualization we can use Chimera or Pyroid. By using Lipgloss and Lipgloss, we can know about the protein ligand interaction.
So now let's discuss about the results. By using cytoscape, which is a molecular analysis tool, we got the top 15 genes which are involved in the laryngeal cancer progression by using the Shastriya expression data set. By seeing the data set, we are gonna see that the top gene is CDC45. CDC45 gene is mainly responsible for cell replication. Now we have done, by taking all top 15 genes, we have done GO analysis. This is gene ontology analysis, which tells the function of the gene. So by the function of the genes, we got to know that the gene is responsible for the top 50 genes are responsible for DNA replication, pre-initial complex reaction. After we have like two classifications of our genes. The second classification we have regulation of transcriptomes of RNA polymerase II promoter in response to the UV-induced DNA damage. After that, we got the gene ontology of the of the top 15 functions. From the top 15 functions, we took the top gene, which is CD605. By taking the top gene, we are going to get the protein from the top gene. Proteins are made DNA is made up of proteins. So we take the top gene, we are going to get the proteins which are in CDC45, and we are going to talk with the suitable ligands to make one molecule. So we have two compounds. We look at two compounds. The first compound, the ligand is binding only at the one side. The second compound will have three binding sites. So if the binding rate is more than 2, it is the most stable compound. The binding rate is less, then distance is binding value. If the value increases, then the affinity also will be reduced. So overall the process, top gene will be CDC45. The main function of CDC45 is cell division, controlling protein 4 by homology. But it plays an important role in cell replication. When the cancer cell is, when larger cancer is in the body, CDC45 is expressed more. The CDC45 expression we got through GAN3 expression analysis. After that, we took the CDC45 and we kept it out, and we got the protein, which is first is 5TGO, which is the crystal structure of cell division protein. By this analysis, we are going to first find a gene which is responsible for the laryngeal cancer. We are going to silence the gene to stop its progression. Conclude this all our research, the gene we found is responsible for the cell division, which is the cancer progression is more because of this CDC45 expression. So we are going to silence the gene by using a suitable ligand and stop the progression.
So that this looks very good. Why you took CDC45? Okay, my question is going for two questions. This company is one. So you do this gene ontology. You found out gene ontology before. So you found out gene CDC45, CDC6, and MCM. And what is MCM?
It's also a gene which is involved in.
So you just took CDC5, CDC45, okay? CDC45 is top gene ontology. What is your size of the gene that you take? So here you have given 14 grand or 15 grand. So what is the size of the gene?
The compounds are more than
Sample size. Sample size 1500.
in what place we have
Correct. Next. So next. So here, where is this UNK? What is this UNK?
the donor is nothing but the protein.
Yeah,
Ligand compound.
I understand what is it, but what is the donor? UNK. UNK. What is this UNK?
The site where the ligand gets binded.
What ligand?
Our protein.
Dog with Pyrex. Okay, UNK dogging. So what is the molecule name? UNK. UNK. What is the molecule name? Chemical ID. There is specification. Okay, come on. Chemical ID. Yeah, I got what you're saying. But what is the name of the molecule? Without knowing the name of the molecule, why you taking dog?
Sir, that UNK is unknown ligand.
See, unknown ligand should have a name for it, right? So benzene is also an unknown ligand.
Actually, we have that binding factor. So it's not
Okay, you see dog. Fine. Fine. Other
we
question.
have it compound, but we did not include it. Actually, it's still ligand factor.
Okay, fine. Okay. So was
Yeah. The first compound has only one binding site. So it's more than number of binding sites, more effective than drug molecule we can attach. More than protein and the ligand forms of complex. More than number of bonds, more strong the bond forms. Nice. Nice. Ligand is like So in the same way, we are going to take the, like, we are going to stop the series in the progress. Actually, this is we are not taking any
Right, I understand. Since it's a computational work, this only one ligand will not.
actually trying to do the compounds. We got many compounds, but the top we got more than
So there is no one. In presentation there is a little bit of problem. Even K, N, what is N? N is nitrogen. Anga, I'll accept that. Glutamine, what atom? Here you say it's N. What atom is N? What atom is in black? That's not atom. N is. So here all N. This is N. N is not. It's not the UNK. N. 1. What does 1 mean? What does 6 mean?
that's one, but like in the outermost shell of electron, they are going to have one electron.
So electron all will not be given in fire experiment.
Like we are with the
What? Hey, you got my point. What is saying? Go to
actually we have done we got the chain component. After that, we utilize pirates. After that, we use that component and everything.
Yeah, I understand what we did, but I just wanted to know what is this.
Actually, by keeping in liquid, we are going to get like 6
Yeah, that's fine. I understand what you did all. I understood what you did. So you did the docking, then you did the plot, you did analysis all. What this means, go to this slide. You should explain me what is this slide is. What is this slide? What this slide UNK, I can understand it's your compound. What does that 1 represent? What does that N represent? What does it not represent?
Actually, it's the outer plane. When you see that
Yeah, because say arginine la. Go to arginine. Arginine A 109 NH1. UNK N103. I can understand DORA. I don't understand the actual. Yeah, that is all fine, man. You should understand the result. Without knowing the result, what I want to say?
actually, our aim is not to gain the multiple stable. We make them to find the system.
Whatever might be your thing, man. You're putting it in the slide. You should know in and out what does this means. Okay. So if someone asks you, you should be able to tell what is it. So what is arginine? ARG is arginine. A is the A chain. 109 is the what? 109 is the amino acid number. NH1 is the ID. Amino acid, not amino acid, is the atom. NH1 that's NH1 atom. Similarly, UNK is your ligand. N is the nitrogen. I don't know what is 1, I don't know what is 3. You should know what is represents. So what is distance? What is the units for this?
Sir, we think that is binding the reason.
I understand what is the link, but you should know what is angstrom. So what is your cutoff range? Above 2, above 10 angstrom is also minus. 2 to 3 is more than 3.2. But 2 to 3 is fine. But why is 3.07 is that? So the distance should be 2 to 5, not 2 to 3. That's what I asked you. So if there is interaction, the distance should be 2 to 5, not 2 to 3. That's why you have added 2.07. There is 2.32. So mathematics would involve mathematics. Normally it is more than 3 should be very good. But if you need to have an interaction, it should be 2 to 5 angstrom, not 3 angstrom. Someone who have thought you in a, so that's what it should be 2 to 3 angstrom, 2 to 5 angstrom. If a atom has to interact with another atom, it should be So 2 to 5 rank. What is your conclusion?
By stopping this, by using the simple drug molecule, by stopping this series for 5 expressions, we can reduce the cancer progression.
Only one drug molecule I have added. There has been lots of drug molecules.
We are just we are not trying the drug molecule.
This interaction also will activate the molecule. How can you say it is stopping the molecule? You are not embarrassing. How we are going to say that it is stopping? So this interaction might increase the activity of the
Further analysis, discussion is like by using the series C4 we can form the drug molecule.
CDC4Y is a good receptor for drug therapy.
We are not doing that. Find the drug we are not doing.
Yeah.
But to add to your research, I would say a small suggestion. You may not clear about what you are going to do. You are giving a drug control. What is your major aim? That objective is
Overtime.
Next is, if you are going to act as an inhibitor molecule in the country, what sort, what types, there are many types of inhibitor molecules. What sort of inhibitor will work exactly here as a clarity?
Major first to have the view of what the gene is, like what is the gene is gene has more expression. What the gene is mainly and in the body, what is its function. After that, do some extra
Okay, so next time do it better with all your
Go on. Be a mic in your for 10 minutes. Make sure that you finish off by 10 minutes.
Good morning, everyone. My name is Govardhan. I am a final year student from Vedic IT Engineering College. And the topic is about the development, optimization, and characterization of dark weak reinforced with texture for effective removal of heavy metal and lead. So, in 21st century, the problem we are facing is both pollution, and my project hopes to address two types of pollution: heavy metal and light pollution. So, as you all know, heavy metal contamination is really a significant problem in our environment due to its toxicity and its putative nature. And light pollution is a very problem for aquatic organisms since it's toxic and toxic. Objectives are three main objectives: to prepare the star scheme and to optimize using response of methodology and to characterize the beach for surface morphology water. So the methodology is simple: start, eggshell and incorporated with 3% sodium nitrate and dropped in 5 calcium. So first it was a trial and error process. 3 types of batches were performed: 10% and 3% sodium nitrate and 3% calcium chloride. So I got the beats were weak and more satisfied. But at the third batch, I have prepared 10% starch and 5 grams of eggshells, and the outcome was strong, and the beats, the eggshells were strongly incorporated. So first, you can see the first batch were weak, not solidified. The second batch, the eggshells are not properly incorporated. The third batch, I got the exact solidified beats, and the eggshells were incorporated. So first I have made two types of beads, S10 E5 and S15 E5. So 10% starch and 15%. So that is what I have. So first I have taken two types of net, and I have taken a net, and I will blue and component. 2 grams of my beach were immersed in this light. And after every 24 hours, the polarimetric assay was done to see the changes in the solution. So these are my results. So you can see the visible color change from before and after solution. So you can see the visible color changes from before and after. And this is the standard graph that I made using the concentration and absorbance method. This is my results. So at the 0 hours, it was around 1.5 absorbance, and the concentration has been found from the standard graph, 300 milligram per liter is the concentration I obtained, and the absorbance is around 2.2 for component, and the concentration is around 300. So after 5 days, the absorbance is reduced to 0.44, the concentration is totally reduced to 27 milligram. So you can see the reduction in the percentage of the metal growth component, and the metal growth was constant reduction, and component you can see after 2 to 20 or 2 days you can see a gradual decrease in the component. So heavy metal removal. So as for heavy metal removal, I have taken lead. Latest state solution was prepared for the same concentration, 300 milligram per liter, and 2 grams of my beads were immersed, same like that, and the remaining lead concentration is identified using 3 nitrogen. So you can see the visible color change. First, before my bead solution, the solutions were cloudy, and after you can see some precipitation in the solution. This is S10 and S15. So after during titration I have taken 0.01 mol of PDTA and 25 ml of my treated lead acetate and 5 ml of ammonium vapor to maintain the pH. Since this is a pH required pH that immediately reacts with the lead. So after eating a solution, the white light color will turn to blue. So that's the time we can identify the volume of so this is the percentage of lead removal. So you can see the S10 E5B, the volume is around 4.5 and the percentage removal I got around 43%. And for S15 E5B, I have percentage removal. So after that, I have done for optimization using response function technology. I have used design expert as well. So these are my table of experiments, and you can see that I have taken 4 factors: starch, eggshell, sodium arginate, and time. And the response I have noted for is metal group, component, and heavy metal. So this is the percentage, and I have noted that 60% removal in metal group and around 45% removal in component and 45% removal in. So these are my table of experiments. So after I have table of experiment, I have went for model validation. So you can see that my model is for metal group I have 80% model is accurate, and for component it was around 92%, and for heavy metal I got 90%. And you can see the standard deviation is around 6.8. So the error value is less in metal group and was very less in the heavy metal. And the critical R-square value is around 68%, and which is close to the R-square value. So my model can be reproduced after. This is the predictive model you can see. So you can see the y-axis the predicted model, and then x-axis you can see the actual result. So the points are gathered around the line. So it is no accurate. So for middle group, this is the unknown table. So you can see the model is very significant, and 2 factors were given as significant, which is starch and time. Since the p value is less than 0.05, but the eggshell and sodium arginate is not as significant. So these are the 3D surface graph. So you can see the color gradient. So blue is the lowest removal percentage of the group, and the red region will represent the highest removal percentage. So the x-axis and y-axis will be different parameters, and the z-axis will always be the metal group. So you can see the changes. So this is other similarities. And since the eggshell and sodium arginate is not that significant, you can see the graph is not right. So response for component. So we can get the same response analysis here. And the 3D graph for metal group, you can see the adjacent removal is the peak of the graph. So but for component you can see the highest removal at the drain, which is at the medium. So these are my curves, and since the sodium hydrogen connection is not good. So for that, so this is the ANOVA table, and these are the so after finding this, I have a bit for optimal configuration. So you can see for start it was around 14.4 grams is urine is optimal, and for HL it was around 6, and the sodium hydrogen it was around 3.5, and the contact time, so you have to leave the solution for around 3 days to get this higher percentage removal, like methyl 58%, component 51, and elemental 47. So characterization, after optimizing I went for characterization, 4 types of characterization performed. So first water absorption capacity. So initial weight and final weight has been noted after immersing the water, and the 300% on average it got around 300% for water absorption capacity, which is good since it will increase the surface area and exposure of the food. So moisture retention. So higher moisture retention keep the mix porous and hydrated, so which will help in the absorption efficiency, improving absorption efficiency. So you can see the after 2 days it was completed. So it is taking 2 days to complete it. So solubility test. So on average it was around 20% solubility. So which is good so the beads won't integrate. So pH stability. So pH stability was analyzed. So initial final weight has been noted, and 3 you can see the slight erosion, and at 11 you can see significant degradation. So the optimum pH should be 95 between 5 and 9. So exactly. So in conclusion, I can say this starch actually. So starch actually we can effectively remove this heavy metal and light. So the response of this method is used to optimize the response condition. And characterization is performed to increase the stability and maintain the heat stability. So this project will help in the environment and sustainability, and usage of waste materials like textile which can be dangerous to the environment. So this is another scalable heat technology which can be used for removal of elements and textiles.
This is great, okay? But this has been already been done. Okay? So you are just doing with starch, Excel and sodium ions. Okay? So why you choose starch and Excel? Excel they are done.
Excel not made that for element. So my raw idea is to incorporate starch and Excel, and effectively it's all to light and
Can you just have you done any experiment only with starch or only with Excel?
I haven't.
Having compared, because I think I have seen papers on sodium acetate, Excels, and starch separate. I haven't seen any papers with this combination. So, but with this combination, have you optimized this combination?
Yes, sir. I optimized this. Yes, sir. I'm using design expert. I have made for cost per method, and I have incorporated these 4 variables of mine, and it has given me the 26 runs. I hope of this 26 runs, and
Another question is, let's say you done the best one, that form these.
Yes, sir.
Form.
Yes, sir. That's why I went for trial and error first. So get the exact form.
Good, but this looks really good.
Okay.
Very interesting. But you should be more confident on the novelty rather than this, because everything is being done. Then whatever experiments you have done, that they done, the call to be done for only single thing, either for starch or for Excel or for sodium acetate. So they haven't combined all these things. That could be your novelty.
Yes, sir. So if the future work will be involving in the improvement of the industry, it was from 45 to 50%. I am really
Thank you.
Thank you.
Shiny extra, that which we call as business. Good afternoon, this is Shiny from Brazilian Engineering College, and I'm going to see the technical project field in CSIR CLRA. My topic is about expanding the genetic code to develop novel fluorescent protein with enhanced properties. And in CLRI, I'm dealing with recombinant proteins, especially with beta sheet proteins. In this, one of my work I explained. Introduction. I'm dealing with fluorescent proteins. These fluorescent proteins will fluoresce in different colors. There are so much of fluorescent proteins like GFPs, RFPs and YFPs. That is green fluorescent protein, red fluorescent protein, yellow fluorescent protein. Among this, I'm dealing with green fluorescent protein, which will emit the light. This green fluorescent protein is derived from jellyfish, which is in aquarium. In underwater, it will glow like blue color while it is present in the water. If we extract the protein from it, it will fluoresce into green due to the specific chromophore structure. There are wide range of applications like thread analysis for fluorescent specific binding emission of animals that are all it is used, and for visualizing cellular levels in antibody tagging. Mostly it is used for tagging with any kind of protein to initially visualize our protein without going for some characterization. So many biosensors, it can be used as a pH sensor to use, since it has color radiation effect. In different pH it will show different colors. Now I am going to explain about green fluorescent protein, which exhibits green fluorescence, which is isolated from jellyfish, and it contains 238 amino acid sequence. This is the structure of beta sheet, GFP, which is a beta barrel. It contains 11 beta strands with it, and this is the structure of the protein. The protein size is approximately 27 to 30 data. Specifically about the chromophore, there is specific 3 amino acids, that is serine, tyrosine and glycine, which is the major chromophore structure which is responsible for the green fluorescence. The specified place of the amino acid sequence will fluoresce the protein, especially with the presence of these tyrosine, glycine and serine. This chromophore of these 3 amino acids will fluoresce. In this work, I am aiming to change the color of fluorescence. That is, I am going to incorporate one unnatural non-canonical amino acid in the place of a substitution of tyrosine. If I am giving any kind of unnatural amino acids in the place of tyrosine, which is the same analog of tyrosine, I can have any one group which will be extra. So I am trying to optimize the condition, and I am going to see how the color changes and how it will be used for certain applications. The strategy which I am going to use is normally tRNA synthase will take the amino acids and it will produce some protein. If correctly the specified 20 natural amino acids, the tRNA synthase will take and it will produce protein. I am going to give my unnatural amino acids, and the tRNA synthase will take these unnatural amino acids and it will produce a continuous protein, which is a recombinant protein with different kinds of tyrosine. For this workflow, which I planned is, I will transform my plasmid into a oxytrophic host strain, and then I will do expression, bacterial lysis, solubility assays, and the specific for incorporating, I need to optimize my strategy. So I firstly transformed my plasmid into oxytrophic strain, and I grow in healthy medium, which is a nutrient-rich medium. After that, I went for adaptation medium, in which 20 natural amino acids will be in the adapting amount. It will not be excess. After that, I will give stress for my cells. After 1 day, after overnight inoculum, I will give stress for my cells, in which my cells will be stressed at least for 20 minutes, in which an excess amount of tyrosine which are present will be washed. Then in depletion medium, I will give minimal amount of my tyrosine, rather than some optimized conditions was there for amino acids. In that, I will give minimal amount
tyrosine constituent, and then after some stationary point, after some hour, it will reach stationary. Then I will give injection and my unnatural amino acid. The bacteria which is craving for tyrosine will take my unnatural amino acid as thinking it is only the tyrosine, and it will take and it will be incorporated. Further, I will do HCl protein expression, and I will purify my protein with FNC column, which is a histone protein column, dialysis and lyophilization. These are the transformed expression of my clones, which I observed after transforming. I observed my plasmids, gene of freeze. Then the confirmation assays. First time is the marker, second time without IPTG with my octotropic strain. Second is plus IPTG. Then after my protein is minus IPTG and plus IPTG. Then optimizing conditions. Actually, in this process, I took totally 4 unnatural amino acids, which is amylotyrosine, fluorotyrosine, DOPA and chlorotyrosine. In these three unnatural amino acids have the same analog of tyrosine. In replacement of some specific in amylotyrosine there will be excess of NH3 group. In DOPA it will be excess of hydroxyl group. Fluorotyrosine it will have the excess fluorine group. Chlorotyrosine chlorine group will be present. I optimize the condition in small scale process, and I observe the color changes when I incorporated amylotyrosine. The E, which is present in red, the GMP protein will change into red. Similarly, while I am giving DOPA, it will change into orange, and fluorotyrosine green, fluorotyrosine light faded. This is some detailed view of my protein, how it is done. First is amylotyrosine incorporating GMP, DOPA, fluorotyrosine and chlorotyrosine. For purification, I use histone column and PLC machine. In that, I purify. The column is also changed like this. In this gene expression, there was a band shift. The fourth link which is present is this image is called amylotyrosine. The fourth link which is present is I gave minimal amount of tyrosine and I induced. Fifth link is that in which I induced my amylotyrosine in the space of tyrosine. So I saw a band shift. There is a change in the size of the protein, as well as after purification also the background band was totally removed and my protein only present. Similarly, it was continued for DOPA and also for fluorotyrosine, also for chlorotyrosine. After that, fluorescent images I took out by using these incorporated proteins. I took fluorescent images. This is the original image of GFP, and this is the amylotyrosine fluorescent image, chlorotyrosine image, DOPA image, and also fluorotyrosine image. After that, I went for doing UV spectrum. In this, I saw emission in fluorescent fluorotyrosine, and I took it for fluorescent spectrum. In that, while taking for fluorescent spectrum, I observed in chlorotyrosine also there was a but in UV spectrum it is not correctly shown. After that, I went for CD spectrum. Since my protein is a secondary protein and beta sheet protein, I checked whether incorporating also there is any change in the beta sheet transfer chain, but there is no change. It showed a beta sheet negative peak in 280 to 250 and the positive peak between 240 to 250, which shows my protein is a beta sheet protein. And I went for denaturation analysis, in which temperature my protein will be stable and it will get denatured. And in specific 70 to 90 degree, my protein is stable, especially for DOPA, fluorotyrosine and chlorotyrosine. These three proteins are stable in high temperature also. So we can take for further analysis, confirm for further analysis. Then I checked for the Maggie whether the size differences are there. There is no size difference. Approximately it is between 27 to 30 kDa. So there is no size mass difference was there. And my future work, I am planning to do biocompatibility assays with this, and also I am planning to do a pH sensor in which normally I am doing, in which I observe GFP and GFP DOPA will completely become invisible under also, if I target my protein, GFP protein to any other protein, there will be a tag. With that, whether my protein is expressed or not, I can see visually rather than going for ACS. So I'm trying to do GFP and anaxin is another protein. I'm trying to tag these two proteins and do some other work, and also that is combination of anaxin and GFP I'm trying to do.
So one question is, work is good, but why you do GFP?
I'm working with beta sheet proteins, not only GFP.
There are lots of beta.
I'm working with SRP12, which is also underwater protein, and similarly spirosil protein.
Because my simple use is say, this GFP is a protein that is used as a tag for breed protein, for fluorescent breeding. So this doesn't have much function in any other thing except for fluorescent protein.
Basically, by incorporating with DOPA, I can see a color change in the protein, which is orange color. The DOPA has a specific property for metal ionic exchanges. So I'm trying to develop a product with the help of GFP DOPA. So in that place, DOPA is an oxidizing agent. If we keep separating the DOPA, it will oxidize. But we found a metal ionic reagent, some in literature reviews, and while we are doing, we found some thing like GFP. So we are trying to develop a metal ionic strategy for GFP DOPA.
Right, fine, that's all fine. But why did you change it to so you can add DOPA to GFP? That's it. So it's going to be green color.
Actually, no. If I am taking DOPA with GSP means it will change into orange color, not in green color. Only GSP will be
So
green
no,
color.
I got it.
Other amino
So another question is, so only the tyrosine that is there inside the beta barrel can change. How we are going to verify? There are definitely many other tyrosines outside the thing. So how we are going to check only that tyrosine can change?
For that, we done some gene expression studies and PCR cloning studies and all we done, and specifically we took one strand of plasmid and then only we transformed it. For the specific analysis, whether my protein tyrosine only is implemented or not, I gave my sample for LCMS, in which I can analyze how much tyrosine amount is there, how much tyrosine is replaced with my DOPA, that all I can check and I can confirm.
But my simple question is, there's no, I would say GFP is just a protein for visualization. Why you need to change that? So there are lots of, so there are green fluorescent protein, there is red fluorescent protein. So there are, so you have done histidine tag. Histidine tag is there. So you have CD8 beats, CD4 beats to tag. So instead of taking a GFP, you have taken a interesting beta barrel. So interesting beta barrel protein.
For one specifically, for choosing DOPA, how do I
DOPA with GFP will give you green color.
it will give orange color.
Correct, but you said it's orange color. But what is DOPA changing the amino acid sequence of GFP?
Only the tyrosine is replaced, but the nature of DOPA is different. If I'm giving
So what is all this mixing up?
optimist whether these amino-tyrosine or fluorouracil having any kind of properties and are having to optimize the conditions are used.
So because to my knowledge, GFP is a very old protein, and for tagging GFP are being used. Now GFP, there is a saying that GFP might interact with protein. So they have taken GFP out. Right now they are going for histidine tag, or they are going for so wherever tags are there. So if you have a very good time and lab, you can go for GFP.
We are rather than purchasing these kind of tags, we normally we are producing these tags and we are going to supply for others also. For that purpose only we are optimizing these conditions with any kind of unnatural amino acids, how it is working.
Because this is too expensive, then I can directly. So what? So your previous expense, what I will do is I have a sequence of my GFP. What I will say is change there and then why you are going
It's not the simple work to do, sir. We need to optimize the condition. If any
So I can
other
make it synthetic. I make it purely synthetic. That is much more easy. So because this is much more expensive for me. So only you know that is going to be the tyrosine we need to take. And synthetically hardly 1,000 rupees what I'm gonna. So I can make this synthetic because this is much more and I need to optimize the condition, I need to change things and it costs more.
we also try for purchasing synthetically, sir, but it costs rather than incorporating this system, it costs so much of amount. We
My
importing.
Still now we haven't concluded that it is only that license getting because tyrosine might have a different function on phosphorylation. So if that will change, where are the
Oxotropic strain, thyroxine oxotropic strain போட்டுருக்கோம். இப்போ அந்த strain நாம போட்டுக்கொண்டு வேற எதுவுமே இது வந்து
That's what my question is simple. So why you are considering this GFP? Yes, GFP is
GFP control
you are taking a different protein. So
different proteins also
that has some functional thing. This doesn't have any function except for promo, except for green protein. This is very much functional.
அதுதான் நம்ம வந்து டோபா போட்டுட்டோம்னா மெட்டல் அயானிக் எக்ஸ்சேஞ்ச் ஆகும். அதுக்காக நம்ம பார்க்கணும். ஃபர்தரா எடுத்து மெட்டல் அயானிக் ஸ்ட்ரென்த் வந்து 2% புரோட்டீன்ல இல்ல. நான் டோபா உள்ள கொடுக்கும்போது தான் டோபா தான் டோபாவோட ஜாயிண்ட் ஆகும்.
Instead of combining with GFP, DOPA let be individual gene, GFP let be individual gene, DOPA does this function, GFP does the function.
அப்போ இது வந்து தைரோசினோட அனலாகும் இருக்கும். தைரோசின் இருக்கும், டோபாவும் இருக்கும். தைரோசினோட காம்பவுண்ட் இது ஜாஸ்தியா
ஏன்
இருக்கு.
அனலாக் போடுறீங்க? GFP நார்மல் GFP போட்டுருங்க.
அப்போ இது டோபாவோட கேரக்டரைசேஷன் எங்களுக்கு கிடைக்காது.
நீங்க DOPA calculation பண்ணல. DOPA what is the function of DOPA in other protein and பாருங்க.
கொலாஜன் புரோட்டீன்ஸ் அந்த மாதிரி புரோட்டீன்ஸ் கூட இருக்கு சார். நான் வந்து அதுவும் பண்ணிட்டு இருக்கோம். நான் மொத்தா 4 டைப்ஸ் ஆஃப் ரீஹாஷிங் ப்ரோட்டீன்ஸ் ஒர்க் பண்ணிட்டு இருக்கேன். CLP, SRP, ஸ்பைடர்சன் அண்ட் GFP. நாலுத்துக்குமே நான் இன்கார்பரேஷனுக்கு ஸ்டடீஸ் பண்ணி இந்த growth factor ஆப்டிமைஸ் பண்ணேன். தைராய்டின் கம்ப்ளீட்டா டிஃபீட் ஆனாதான் என்னால இந்த டோபாவோட கேரக்டரைசேஷன்ல புரோட்டீன்ல அனுபவம் முடியும்.
With your research, I am not convinced that it is that tyrosine gets changed. Because there might be other tyrosine. That might also get changed.
என்னோட ஜீன்ஸ் ஸ்டாண்டே நான் இப்போ தைரோசின் தான் இருக்கணும் அப்படின்னு ஆல்ரெடி க்ளோன் பண்ணி ட்ரான்ஸ்ஃபார்ம் பண்ணி வச்சிருக்கேன். ஆனால் அதனால எனக்கு கண்டிப்பா தெரியும் தைரோசின்
So you know how many
மாலிகுலர்
tyrosines are present in your feed? 62 tyrosines are there. So 62 tyrosines. So I don't know what tyrosine you are going to obtain. What happens to the other 61 tyrosines?
தைரோசின்ஸ் மீன் டோபாவா மாறிடும். அதை நான் எப்படி விஷுவலா நான் பண்றேன் அப்படின்னா நான் இன்னொரு ப்ரோட்டீன்ல ஒர்க் பண்றேன். அதுல தைரோசினுக்கு வந்து அமினோசைட்டோசின் கொடுக்கும்போது கம்ப்ளீட்டா தைரோசின் டிப்ளீட் ஆகும். அப்படி ஆகாததை நான் எப்படி கண்டுபிடிச்சேன்னா ஜெல் ரன் பண்ணும்போது எனக்கு டபுள் பேண்டா தெரிஞ்சிருச்சு. தைரோசின் நார்மலா அந்த பேண்ட் ஷிஃப்ட் நான் சொல்றேன். அந்த பேண்ட் ஷிஃப்ட்ல ஒரே டபுள் பேண்ட் இருந்துச்சு. ஆனா இந்த டோபாவெல்லாம் நான் இன்கார்பரேட் பண்ணும்போது அந்த டபுள் பேண்ட் தெரியவே இல்லை. சிங்கிள் பேண்டா தான் இருந்துச்சு. அதுக்கு நான் என்ன கன்ஃபார்ம் பண்ணிக்கிறேன் என்கிட்ட இருக்கிற 62 தைரோசின் மாலிகுலுமே 62 அன்நேச்சுரல் அமினோ ஆசிட்ஸா மாறிடுச்சு. அது இன்னும் ஃபர்தரா நான் எஸ்டிமேட் பண்ணிக்கிறதுக்காக தான் எஸ்சிஎம்எஸ் நான் கொடுத்துருக்கேன். கன்ஃபார்மா என்னால தியரிட்டிக்கலா இப்படி ஒரு அப்ளை பண்ணி நான் கண்டுபிடிச்சிட்டேன். என்னோட ப்ரோட்டீன் எல்லாமே மாறி என்னோட தைரோசின் எல்லாமே மாறிடுச்சு அப்படின்னு நான் கண்டுபிடிக்கிறதுக்காக எஸ்டிமேஷன் கொடுத்துருக்கேன். அது மூலமா என்னால கண்டுபிடிக்க முடியும்.
Next. The next one. Very good.
So what happens is, normally we know that we have only X and Y, right? So these things, they have XY, meaning there's an extra X chromosome that's going to stop the effect of the Y chromosome. So the picture pieces of people with Klinefelter syndrome include hypogonadism and mainly infertility. This is one of the most important side effects of this. So the problem with Klinefelter syndrome is underdiagnosis. So most of the cases, around 70% of the cases, underdiagnosis. So the main purpose of this project is to identify markers that are expressed within the disease. So the approach of taking is the transcriptome approach. So RNA sequencing is done with Illumina and next-gen sequencing techniques. And so this project is going to take data sets that are publicly available and identify what they are markers. So this is actually work in progress and not completed. So I'm going to share the progress in that program. So the objective of this project is the identification and collection of the gene expression data from public databases. So geodatabase is where I am it. Next is security appropriate tools for bioinformatics management. And also performing the bioinformatics management to identify the markers. So this is the workflow of the project. So the workflow corresponds to microdata data, my data with standard sequencing data. So the data analysis will not use Sigma but rather use RNA. So using RNA we can identify all the different expressed genes, and based on what is differently expressed, we can do a lot of other things such as gene ontology, identifying the protein-protein interaction network, and also the histone network. So the artistic workflow that I followed is to get the data in Illumina sequencing. Then FastQ format, files were done for quality control. So I used FastQ as a tool. Once quality control is performed, I went on information. I took all the data sets are very low quality. So by doing it, only the data sets are high quality and also the low quality data sets converted to high quality data. So once we've done everything, quality control is an option that takes the step is not sufficient. Then we go on mapping. So things to the transcriptome, we can use tools like both things. So we can identify what are the genes that are expressed and what are the positions for the throughout the human chromosome. So we can go on to quantification. So we use count. So how much the gene is expressed, how many counts are expressed in the control versus the Klinefelter patient. So once you get quantification, once you have the actual numbers, we can go on different expressions using CQs and R tracking. So we can identify a lot of the data we got from chromosome, and we can move on to protein genealogy. So one data vector is the exact KB ID that I've used from the geodatabase. So Klinefelter syndrome, there are two cases and two indications, and there was one indication. So the reason I chose this is because there are two applications for both users, Spermid and Klinefelter syndrome. So this is my workspace that I use, the tools that I use. I use MAPA, and also I have fast vision, ISAC tools, and so take out that data. This is the pipeline. I did fast vision first to get fast vision report. Then I moved on to the data. Then once I have all the information, I went on the ISAC tool to build the initial genome of the human. And based on the built genome, I went on to take a count. So that's what we count. So this is based on the initial part of the report. So this represents one of the bases that have a quality. So we can see the quality score over the scope. So most of the bases that are very recent, very low quality. So for both Klinefelter patient 1 and patient 2, a lot of bases are actually running in this form. So once we treatment, that a lot of the patients have moved to the brain region, which is actually what we do for patients. Once we obtain the I went on to ISAC to build the reference genome, basically the data to the reference genome. So once the files are built, I went on to gene count. Not very significant, but you can see that there are approximately 60,000 genes, and these genes represent how much of it is expressed in the Klinefelter system and how much of it is controlled. So we can do the comparison. So
So far, this is where I'm finished. So based on this, if you want to read into that, you can exercise the log code values and also Arduino blocks. See what is the different states to see and what acts as biomarkers. So you can identify biomarkers. If you can move on to therapeutic aspects, like see what kind of drugs can be used for targeted biomarkers. So this is really important, and I aim to mention another two or three. Actually, the data that I collected goes from around 50-year-old people. So, yeah, because all the sexual characteristics only develop after puberty, post-puberty. So, to identify the pre-puberty, we have to genomic analysis. So, we can identify that there is an extra chromosome. But the main point of this is for those people who are born, so if someone is already born, we can't go and specifically change all the remove the chromosome for peace. So for those cases, to reduce the adverse effects of the underlying conditions, we want to identify the biomarker. Yes. So we can identify it more work. So we can do it naturally. Now, if it is possible, actually, some of the cases they pass through, even though they don't show up the test, the patient may to be affected by cancer. So, for those, they may be affected by cancer, even if the test shows like that. So, that's false positive. Yes. That depends on the test itself. Like maybe there were some contamination with the fluid that is involved. Also, it happens, mistakes happen. What the problem happens, what happens is, once the individual is born, we cannot reverse that all this is happening. So for those things who have been affected, we want to identify biomarkers so we can treat the person. Yes, these were taken from the test of 3-year-olds. So this was done transitory. Yes, I understand. But the issue with that is like we can only take the samples when that person is there. So human data, we cannot really go for so we can go inside of studies and for mice and rats. But that's the only alternative if you want to look at different states, pre-puberty. If we have someone who was of under age 30 and who is not alive, what is willing to join the office, or then we take their data. Otherwise, we can't get it. So, I went to like May around 2030, and most things were opportunity because those are the age that people have. So, you can't get it. I understand your point, so like my point is, if someone is affected by time business, there's nothing to do to reverse it. So we have this is a single page solution. So we can go ahead and do bone marrow replacement with the current system, and furthermore they won't have any issue. But the thing with mitosis is we can't change like all the cells of the human body. So this is served only those who are affected by time business. If someone is not affected and they can identify pre-birth, then all is good. The parents. Yes. Can you reduce the percentage? Yeah.
So you can work on that part.
Even 3%, we can't do anything.
If it is possible, that's what I'm asking. If it is possible, you can work on it, we can add more.
Okay. So far, I don't think that happened before.
Yes, that's what I'm telling you. That will add real innovation to your research.
Okay.
incoming workflow, the name and